All right. Good afternoon, good evening, and good morning, everyone, depending on where you might be. I apologize for this late start in our BioTalk webinar series. Unfortunately, we had some technical difficulties. I am Rob Burgess, and I head up business development for Sino-Biological. And I want to welcome everybody to the next installment in our series. We have a wonderful speaker by the name of Dr. Hua Wei, based down in Houston, I believe. And he's going to talk to us about advances in antibody drug conjugates. Before I officially introduce our speaker, I just want to mention one housekeeping issue, and that is we ask everyone, if you have a question, to please put it in the chat box. And following the seminar, I will run through the chat box questions and get to as many questions as possible and have our lecture person actually answer those questions. So thank you for your patience there. We'll withhold all questions to the end. Also, if you get a chance, please put in the chat box, say hello, and tell us where you're from. We're always interested in the different regions and countries that people are calling in from. So with that, it's my pleasure to introduce to everyone Dr. Hua Wei, our speaker for this afternoon. Dr. Wei is currently Vice President of Industrial Clients in the Department of Shanghai Model, or Vice President of Industrial Clients Department in Shanghai Model Organism Center, which is a subsidiary, which also has a subsidiary known as Genobiotics, if I pronounce that correctly. And he holds the same title at Genobiotics as he does at the Shanghai Model Organism Center. He received his Bachelor and Master of Science degrees from China Agricultural University. And in 2008, Dr. Wei received his doctorate in biochemistry from The Ohio State University. In 2009, I believe it was from about 2009 to 2012, Dr. Wei performed his postdoctoral training at MD Anderson Cancer Center in Houston. And also in 2012, Dr. Wei served as a principal investigator at the National Human Genome Research Center in Beijing, China. Subsequently, he joined several biotech and biopharmaceutical companies, such as Gloria Biosciences and Ardavila Biopharma as preclinical director and senior preclinical director. He was responsible for the preclinical research on antibody and small molecule drugs, and one drug there was successfully approved in 2022, and another was approved for clinical trials by the NMPA in roughly mid-2022. In mid-2022, he formally joined Genobiotics in Houston, Texas. And the title of Dr. Wei's talk today is Advances in Antibody Drug Conjugates, Development and Preclinical Models. And so I'll turn it over to you now, Dr. Wei, and it's an honor to have you this afternoon.
Okay, thank you, Rob. Thank you very much. And thank you, Sino-Biologic team, so for this great opportunity to me to give a presentation to our audience. So the title of this presentation is Advances in ADC Development and Preclinical Models. So this whole presentation consists of 3 parts. The first is introduction of ADC. The second is the ADC preclinical models. And the third, I will talk a little bit about the challenges and the prospects of ADC. So the first is the introduction. So ADC, the full name is antibody drug conjugates. It consists of 3 parts. First is the antibody. The function of it is to recognize the tumor-associated antigen and basically to bring the drug to the tumor. And the second important part is the linker. It stabilizes and also brings the toxin, also called a payload, to the tumor sites. The third part is a cytotoxic drug, which is very potent chemicals. The function of this drug is to inhibit the tumor and also kill the tumor cells. So why the ADC is so popular right now? Because it's a powerful therapeutic tool to kill tumor cells while minimizing damage to healthy tissue. And the the market analysis data showed that from 2024 to 2028, there's about 23% of increase, annual sales increase each year. And also in last year, 2023, totally 76 ADC deals made, including license, collaborations, and acquisitions. The total amount of these deals exceeded $100 billion. And also, this is just the beginning of the ADC drug market. So I will talk a little bit about the history of the development of ADC drugs. The original concept was proposed by Paul Ehrlich in 1910, about 100 years ago. He proposed a magic bullet that can kill the tumor cells and do not damage the healthy cells. And in the year 2000, the FDA approved the first ADC drug, but later, due to its severe side effects, it pulled out the drug and upgraded to the second generation and then get approved again later. In the year 2013, so the one representative of the second generation of ADC was approved by the FDA. I will talk about this in a few minutes. And in 2019, a third generation of ADC was approved. And also I will talk about this and to see the difference between second generation and the third generation ADCs. So totally, there are about more than 100 ADC candidates are in clinical trial. So if we currently, FDA has approved 15 ADCs for solid and hematologic cancers. If we have a close look of these approved 15 ADCs, we can find out that most ADCs use the clearable linkers, and most of the linkers are use the enzymatic activity to cut the linker and then release the toxic drugs to kill the cancer cells. And most common payloads are DNA damaging agents and tubulin inhibitors. But right now, the DNA damaging looks like more potent than tubulin and a little bit more popular currently. So this slide shows the mechanism of action of ADCs. So it's quite complicated, and it includes 6 steps. But I won't, because of the time constraints, I won't go too many details of these mechanisms. But if you are interested, you can go to the read the paper on the slide. And basically, 6 steps involved, like ADC circulating after the ADC drug injection into the blood, and then they will the antibody will recognize the antigen on the tumor cells, and then the tumor cells will initiate the internalization and then bring the ADC drug into the tumor cell, and then the endosome will lysis, will fuse with the lysosome, and then the enzymes in the lysosome will go into the endosome and then cut the linker using its enzyme protease activity, and then the release the toxin can go to this target and bind to the target, and then finally inhibit the tumor growth, and also to induce the apoptosis of the tumor cells. So because this is a complex procedure, so each step is critical for the ADC's function. So also these all steps shows that it's very hard to get a very good ADC drug. So there are 2 if the ADC drug has the isotope, the antibody's isotope is IgG1, so it has 3 actual FC-induced functions like CDC, ADCC, and ADCP. This this function can also contribute to the cancer cell killing effect. But the later part, later two part like ADCC and ADCP, can also influence the drug's PK effect, like half-life in the circulation. So usually people do a modification of the FC part and then to reduce the ADCC and the ADCP effect in order to increase the half-life in the circulation of the ADC drug. Also, if the target is oncogene like HER2 or EGFR, the ADC, once ADC bind to its target, they can also block the ADC's homogenization or bind to this ligand or the other partner to further inhibit its signal transduction pathways to further increase the cancer killing effects. So also, if we have a close look of currently more than 100 ADCs in clinical development stage, in each category, we can find out easily find out that the ADCs are pretty focused on the top 3 candidates of each category. More than 80% or 60% of the ADCs are focused on like just the top 2 or top 3 candidates, which means there are a lot of space for the future ADC development opportunities. For example, we can find more target or use more carriers to optimize the ADC, but definitely it needs a lot of preclinical experiments to reveal the optimum function of the ADC. So this is the representative of the second generation of ADC. The drug is called trastuzumab enhancing. It's also called TDM1. This ADC is developed by Genentech. The DAR drug antibody ratio is 3.5, which means on average, there's 3.5 drugs linked to each antibody. And the linker is MCC linker. It's a non-clearable linker. And the drug is DM1. It's membrane non-penetrable, so which means that this ADC has little or minimum bystander effect. I will talk about this bystander effect later. So the ADC, as I mentioned, this drug was approved in 2013 due to the global phase 3 trial. So compared to the conventional chemotherapy, not only the PFS and the ORR are much better than conventional chemotherapy. Here is another example. This is the third generation of ADC. It's called Enhertu. So it is developed by a Japanese company called Daiichi Sankyo. The DAR drug antibody ratio is 8. This time it improved the linker, used the clearable linker. And also the DXd drug is more potent than the DM1. And also this drug is membrane permeable, so it can go to the other cells around the tumor cell and then kill the, especially those tumor cells with low expression of antigen. So it has a strong bystander effect. And later the clinical study shows much better cancer killing effect than the second generation of ADC, than the previous one. So, and there's 7 key attributes of this DXd ADC, like a payload, shorter systemic half-life, which means the toxicity is lesser compared to the second generation. And here is the head-to-head clinical study comparing the Enhertu with the TDM1, the first antibody ADC I mentioned. From here, we can see the PFS is 28.8 of the Enhertu compared to 6.6 of the TDM1. So the reason why it has so large difference is due to the bystander effect, more higher DAR, and the clearable linker. So which means that to design a good ADC, so we need to consider lots of aspects. So the later clinical study showed that the Enhertu showed excellent efficacy in patients with low and even ultra-low HER2 expression, which means this drug can cover nearly 90% of HR-positive breast cancer patients. And most recently, the FDA approved Enhertu as the first tumor agonist HER2 directed therapy in multiple solid tumors, like cervical, endometrial, and ovarian cancers. So due to its very good clinical study results. So second part, I'm going to talk about the ADC's preclinical models. So how to develop the good ADC? So because ADC is so complicated compared to the conventional antibody drug, so multiple aspects should be considered. So first is to choose the antigen, like tumor-associated antigen, which can determine the ADC's specificity. And also, when we choose a good antibody for the ADC, we need to consider the internalization, and also we better use the humanized antibody to reduce the immunogenicity of the patients. And also, we have to remember that we need a long half-life of the antibody. And also, currently, most popular ADC are using IgG1 isotope because it has a long half-life, and some of it has the optimum FC function. And also, the second part is to choose the good toxic drugs. Currently, most popular toxic drugs are tubulin inhibitors, and the second is the DNA damaging agents like SN38 and DXD. And currently, most popular DNA damaging DNA damaging agents are the most popular right now. And also, some companies are using the modulators like a TLR agonist and a STIN agonist to increase the NK or T cells functions. So in terms of the linkers, right now, pharmaceutical companies are using more and more clearable linkers because these linkers has a strong bystander killing effect. So it will enlarge the toxic drugs, drug killing effect to the low energy expression cancer cells. And the most popular clinical cleavable linker are the enzyme cleavable linker. So lastly, the conjugation method is also important. It can increase the drug to get the optimum drug antibody ratio. And right now, most popular method is the site-specific conjugation method using such as engineered reactive cysteine residues, glycoside-specific ADCs, and then non-traditional amino acid to modify the amino acid, and then use the amino acid to conjugate the toxic drug. and also use a dicysteine bridging method to introduce the toxic drugs into the ADC. So there are lots of methods to evaluate the efficacy of the ADC in vitro and in vivo. For example, the targeting validation and affinity measurements, we can use flow cytometry, ELISA, SPR, and BLI to characterize the ADC's property. And also, we can use the hydrophobic interaction chromatography HPLC to characterize the DAR to determine how much the DAR value. And in terms of the in vitro functional study, so it involves ADC internalization, which can be done by FACS, confocal microscope, and in vitro cytotoxicity analysis. We can use the cell variability, bystander killing. The effect can be done by the InQSite live cell analysis system, and also apoptosis and cell cycle kit can also be used to detect the ADC's cytotoxicity analysis. And also we need to characterize the FC cytotoxicity function by ADCC, ADCP kits. So to perform the in vivo efficacy study, we can use a CDX model, PDX model with the IVIS system to see the tumor grow in vivo tumor cell grow in the mouse body. And also we can target the profile in PDX models like using the fish or IHC to characterize the tumor growth in mouse models. And finally, we also need to use the PK to test the ADC's PK, PD, also toxicity using different methods like ELISA, MSD, and HE, and also use the IVIS IVIS system to detect the distribution of ADC. And later, if we want to apply for to submit the IND application, we also need other experiments like thermal stability, integrity analysis for the ADC drug. Yeah. So I will give an introduction of one paper and to show the recent progress in the ADC drug development. So basically, they use the phage displayed antibody library, two different libraries. One is FAB, another one is single domain antibody library. To use these two libraries to screening human oncofetal antigen 5T4, which is a tumor-specific antibody, specific protein antigen. So they use this library to screen the antigen and find out two drugs. One is the conventional antibody 603. Another one is the single domain antibody, which is called 501. And then they use the conjugation kit to link to use a linker and conjugate it to SN38, which is the DNA damaging toxic drugs. And then finally, they got the two different ADC. One is called 603 SN38. Another is 501 SN38, two different antibody. The DAR of the first one is about 8. The second is 1. So first they tried about 8 different tumor cells to see the antigen level, and the results showed that from the high to low level. So they choose the 3 different antigen expression cell lines to do the cell viability test of these two ADCs. So results show like B. So both ADCs can inhibit the high expression of cell lines significantly, and compared to the medium and the low antigen expression cell lines. And the second, they used the dye-light 650, which is a dye to label to these two different ADCs, and then incubate with the cell line. And then after 4 hours, they find out that the 603 has the higher internalization compared to the single domain ADC. So next, they tested the ADC affinity for both proteins and for different cell lines. So what they found out that the low expression cell lines has the lowest binding of the ADC, of both ADCs, and also the ADC conjugation, the toxic drug conjugation, SN38 conjugation didn't decrease the antibody's binding compared to the parent antibody. And then next, they used the two different experiments to test the tumor tissue penetration of these two ADCs. The first one is pancreatic organoids. The second one is spheroids of the BSPC3 tumor cells, which is the pancreatic cancer tumor. So after 24 hours incubation of ADC with the organoids and also the spheroids, so what they found out is the single domain ADC has the much stronger tumor tissue penetration ability compared to conventional like 603 ADC. So next, what they did is they used the test the mouse inoculated with tumor cells, and then give the one dose of both ADCs, and then to do the time course of the ADC's distribution. And then what they found out is after just 1 hour after the drug injection, the single domain ADC peaked at the highest radiance. But however, compared to the conventional ADC 603, the highest concentration is much lower compared to the 1 hour of the single domain ADC. So which means the single domain ADC exhibits a rapid distribution to tumor sites and a higher tumor uptake. And the figure C shows the PK data of both drugs. So the results shows that the single domain ADC has a significantly shorter half-life compared to the 603 conventional ADC drug. So finally, they did the in vivo efficacy test. So after sub-Q inoculation of the tumor cell lines, BXPC3 cell lines, and then they give different doses of these two different ADCs, or the same dose but different dosage. One gives 8 times, another one gives one is 6 times, another one is 4 times, because the single domain ADC has a shorter half-life, so they give 2 more dosage of the single domain ADC drug. So the results showed that the single domain drug ADC has the highest TGI compared to tumor growth inhibition compared to the conventional ADC drug. So finally, the conclusion shows that a single domain ADC showed deeper tumor penetration higher tumor uptake and faster accumulation at a tumor site than conventional ADC, and resulting in improved tumor inhibition. So we have lots of CDX and PDX models. We can provide biopsy, nude mice, North mice, or even the immunodeficient mice to meet the ADC preclinical study needs. And also we have more than 20 different PDX models, which can be used for the ADC drug evaluation and also drug development. So currently, to support the drug development, antibody drug development, and also ADC drug development, we have developed more than 800 different humanized knock-in mouse. All background are C57 biopsy or C57 BLC6 background, but we have other backgrounds like rats biopsy background and even the AMG background. And these are the mouse models we generated for the popular ADCs. So you can go to our website to check the validating data and all related mouse informations. And also, as I said, we have the MG mouse model, which is the highest degree of immunodeficiency and also long lifespan. It's ideal for the xenograft model with minimum immune ejection to support the ADC development, especially for the combination development like ADC combined with chemotherapy, combined with ICI drugs like PD-1, PD-L1 antibody. So, and also we can, this model is perfect for the human immune reconstitution, like PBMC or the CD33 HSC engraftment experiment. And this mouse line has been validated for more than 200 human cancer cell lines on this model. So besides the mouse models, we also developed more than 500 different humanized cell lines. Most of them has the in vivo efficacy data to support the drug development, like antibody, bispecific antibody, and ADC drug development. And this is some cell lines for the currently popular ADC drugs. And also to do the in vivo ADC efficacy test. So we also developed about 100 different luciferase fluorescence tracing cell lines for the in vivo in vitro studies, especially for the bystander effects test and also in vivo ADC drug for the in vivo ADC combinational drug efficacy test. Also, we have lots of human cancer cell lines can be used for the development. So this is the one example of our of the ADC bystander killing effect. This experiment was done by our company. So basically, we use the TDSD to treat the high expression, low expression antigen expression, 2 different cell lines. For the first cell line, SKBR3 cell line, because it has a high expression of antigen. So the antibody, the ADC showed optimal effect. But in terms of the low expression antigen, low expression cell lines, the ADC does not show any effect of inhibiting the cell growth. But if we incubated these 2 cell lines together with a high expression, low expression cell line together, and after adding the ADC TDXD, so even the minimum concentration of the ADC has strong strong cytotoxicity effect compared to the non-ADC vehicle only. So also, we also tested that the combinational effect of the ADC using our humanized cell lines. So basically, the ADC plus the PD-1 Keytruda shows the highest, strongest tumor cell inhibition in vivo, which compared to the parent ADC JAK only. So here's another effect. So the use humanized C57 mouse models. After inoculation of the MC38, HB7, H3 tumor cell lines, and then after given the drug ADC with the Keytruda effect, the group 6 shows the highest TGI compared to the other groups. So which means in the future, so the ADC drug may be combined with the PD-1 drug to achieve higher clinical benefit for patients. So another one, the bispecific antibody. So we tested bispecific ADC on our mouse models, and also the results showed that 4 mg per kg bispecific antibody achieved the optimum TGI, tumor growth inhibition effect, compared to the NDC only. So last part, I want to do a brief introduction of the challenges and the prospects of ADC drugs. So the largest challenge is the limited number of targets for ADC. Right now, there are more than 100 ADC in clinical stage, but most of them are focused on just less than 10 targets. So these targets can be assorted to different categories, like the first one is target antigens always expressed in cancer cells, like PSMA, TF, and CD56, also 5T4, as I mentioned before. And also there are some targets in the hematological malignancies like CD22, CD30, CD74, etc. And the oncogene targets, like HER2, EGFR, and some target is expressed in the tumor vascular system like ETB, PSMA, VGF2, etc. Tissue factors, etc. And some targeted are in the matrix like collagen 6 and the tenacin C, etc. So another obstacles of the ADC is the resistance to ADC, because the tumor cells usually express only single antigen, and which brings the heterogeneity. Some tumor cells are sensitive to ADCs, some tumor cells are resistant to ADC because of the low expression of antigen. And also after treatment, the tumor cells can evolve to reduce the antigen expression, and also down-regulate the antigen expression of the tumor cell membrane, and also even like internalization of the antigen. And some part of the tumor, the mechanism is due to the epitope mutation, and to activate the bypass pathways. All of this leads to the ADC resistance. And also the tumor cells can express MDR, like multi-drug protein, can efflux the toxin to outside of the tumor, so the tumor can survive. So to fight against the resistance, So new strategies are required to fight the resistance, such as better payload or use different payload and better linker, or even change the antibody. And also, combinational therapy is another method to fight against the resistance. And also, the biomarkers are needed to monitor and discover ADC resistance in the early stage. So another trend is to develop the bispecific antibody. So recently, BMS just agreed a 8.4 billion ADC contract with Systel Immune company. So which is the bispecific and ADC antibody and ADC drug. One end recognize the EGFR, another end recognize the HER3 antibody. So because one antigen has higher internalization effect, so with the internalization effect, effect is much better than the parent antibody. So it can overcome the drug resistance, therefore increase the drug safety, because the ADC needs to bind two antigens to initiate the killing effect. So which reduce the side toxicity of this drug. However, to develop the bispecific and ADC drugs, we need to focus on the antigen selection and also optimization of ADC's epitope binding pattern and affinity. These three remain a challenge because it's very hard to find two antigens can have the 1+1 larger than 2 effect. So finally, the future prospects of ADC drug development. So first, we need to reveal the relationship between ADC structural components and their PK/PD. And the second is as I show in the paper, so small size nanobodies can be used for tumor penetration to improve efficacy. However, the short half-life limits the application of nano antibodies. So the third is there are many things remain to be improved, like in vivo stabilization, like hydrophilicity of the drug and the antibody, and also site-specific conjugation. We need better site-specific and conjugation to better the homogeneity of the ADC, and also to increase the efficacy. We need sometimes needs higher DAR, sometimes highest DAR is not optimum because it decreases the PK of the ADC. So some of the preclinical study should be done to get an optimal ADR of each specific ADC drugs. And sometimes we can change the toxic drug, use the dual payloads, and also use the combinational therapy like IO drug, and also like BS bispecific antibody. And also important aspect is to reduce the toxicity. Like we need to find more tumor-associated protein, and also to find the tumor site-specific payload release. And also the fourth is to better understanding of molecular mechanism of action of ADCs, like a targeted delivery, antigen internalization mechanism, and combinational therapy mechanism, and also new conjugation sites, chemistries, and linker technologies are needed to develop and also to improve ADC's efficacy and to reduce toxicity. So finally, most importantly is extensive preclinical and clinical studies are necessary for a good development of ADC drugs. Okay, thank you very much. That's all my presentation. And please contact me if you have any questions or need our help for your project or for the product. Thank you.
Great. Thank you, Dr. Hui. That was an excellent presentation. We appreciate your time putting that together and presenting it to us. Now, I would just remind everybody that's still online to please, if you happen to have a question for Dr. Hui, just type it into the chat box and I'll be sure to ask it. I'll go ahead and start, Dr. Hui. I've got a few questions of my own, and please forgive me if you answered these and I just missed them in your talk.
Okay, no problem.
The first question is, for an antibody drug conjugate, is there the possibility or the evidence that exists that maybe an ADC has more toxicity than if you were to administer the monoclonal antibody and then the chemotherapy or the drug, the small molecule drug, separately?
Yeah, I think so. Definitely ADC, because it carries much more potent toxin. So it's very important to link the toxin with the antibody, but the antibody can still has like off target or even the low expression. Normal cells can express the tumor associated proteins, not the tumor specific, although tumor specific antigen is the preferred target. But yeah, sure, it has more toxicity than mono antibody and or even the chemotherapy, because the toxin in the ADC is much more potent than normal chemotherapy drug or chemicals.
Right.
Yeah, but as I said, we can use bispecific antibody, bispecific ADC, which can more specifically recognize tumor cells. But again, yeah, I think the benefit is larger than the risk. So that's why the ADC is quite popular right now. Yeah.
Right. Great. Thank you for that answer. Just to remind everybody on the call, Sinobiological does design custom bispecific antibodies, which fits, I think, very nicely with this talk.
Okay, great. Thank you.
Just had to throw that in there. Also, and again, maybe you mentioned this, but other than tumors and cancers, malignancies, and so forth, are there any other areas of disease or human physiology that ADCs might treat or benefit?
Yeah, I think this is a very good question. So I think maybe currently it's not very hard right now, but maybe in a few years, the company are doing this secretly. They won't reveal the knowledge to us. But I think it should be like there's a promising field in autoimmune disease, so which the ADC can be used. But as I said, we need to optimize the dose and optimize the linker and the drug to develop to get the optimum ADC drug.
Yeah. Okay, great. Thank you for that. And while we're waiting on any other questions that the audience might have, just one more question I have. To me, it's a little unclear what the offering is of Genobiotics, and I think this is an excellent opportunity for you to get the word out about the products and the services regarding the animal models to the community. So can you just summarize very briefly what is the business model of Genobiotics and what are the animal models / products and services that you provide? Yeah, so as I said, we Genobiotics has lots of mouse models, humanized mouse models, and also we provide a CRO service for lots of pharmaceutical companies, and then do the even develop the mouse model if they do not have, and also we have more than 20,000 different mouse models, so which is very convenient for the researchers or for pharmaceutical companies, because to develop a mouse model, especially like knock-in, knock-out mouse model, it takes at least 1 year to do that. So pharmaceutical companies are time sensitive.
So if once they get the mouse model, they can start the test, the drug test, the efficacy test in vivo, in vitro, and very quickly. So which means a lot to those pharmaceutical companies.
Yeah, so you do knockouts and knock-ins there, and you do tissue specific and ubiquitous, I guess, right?
Yeah, all of them. Yeah, because our company was established in 2001, so we have more than 20, about 24 years of history. So
Right.
so during this period, we accumulated 20,000 different mouse models.
Yeah,
So
that's great.
Yeah.
Okay. And CRISPR-Cas9
Yeah.
based, I guess, genetic engineering. Okay.
Yeah.
And,
Most of cell based CRISPR technology based mouse line or cell lines. Yeah.
and I assume you don't have an animal facility that houses all of those 20,000 models, but you have them frozen back as embryos?
Most of them, maybe half of the, maybe how to say that? Some of them are live, but some of them are embryo or sperms. But we can derive it very quickly in 3 months. Yeah.
Right.
Yeah, because it's a huge cost if we keep all the mouse alive. Yeah.
Yeah, I used to be in the knockout business, so I know about that cost.
Yeah.
That's great. Well, I think we'll wrap it up there, Dr. Wei. Again, that was an excellent seminar. A few folks I want to thank. I want to thank the attendees that joined us today. We had a pretty good turnout. Wish y'all would have asked questions, but maybe next time. Certainly, Dr. Wei and your colleague, Jane Hong, I appreciate your time today. Again, it was quite an honor to have you give this talk, and it was a wonderful talk, very informative. I know a lot more about ADCs now, and I'm sure our audience does too. And congrats to you and Jane on your success with Genobiotics and the Shanghai Model Organism Center as well.
Okay.
And then finally, one other person, Mr. Clayton Miranda, who's my friend and good colleague at Sino. And he's the one that actually administered this webinar today. So Clayton, thank you for your efforts. And with that, I guess we'll sign off and say good night or good afternoon to everybody on the call. Thank you.
Thank you, Robert, for giving me this opportunity to give the presentation. I really appreciate that.
Thank you.
Thank you very much. Thank you, Simon.